Hello and welcome back to this online NPTEL certification course on aquatic geochemistry. And this is your 10th lecture, and as we were discussing, the water sampling techniques, we will be discussing in this lecture as well. So in the last lecture, we were talking about, how the river chemistry varies across the spatial scale due to different reasons, and that we have to be thoughtful about all these different parameters while we are going out for sampling. How about for oceans? Now, as you can see in this particular plot, the height of the ocean is given, and you can see that there is a variation in from in for a few meters. Okay, so that is due to salinity, that is due to density differences, that is due to the thermal heating and all that. So therefore, as you can see, there are ocean currents being created, and they are continuously moving. So when you're for as I already mentioned, when you're having an ocean expedition, they will be of course taking into account whether you are trying to follow one particular ocean current, whether you're seeing, if you're taking one current, whether that is changing, okay, how well it is moving and mixing, okay, mixing with the other ocean current, or there is certain climate zones you're crossing, or there is local stressors. So there may be a huge port here that is creating polluting, there may be plastic pollution or something is changing. Okay, so there can be other parameters. Okay, same thing for groundwater. So in groundwater, as I have already said, you cannot see on the surface. So what you have to do, wherever there is wells, you can see how the aquifer is moving. So if you're trying to find out pathways, that is the water pathways, you can use tracer based experiments and all that to find the pathways. If you're trying to sample the deep groundwater, okay, that is this one, the confined aquifer. Okay, so the deep groundwater generally is free from a lot of shallow water contamination, like say suppose if there is an agricultural field, there's a lot of fertilizer application, then it can easily leach out nitrates or sometimes metals like lead. Okay, it can leach out those elements into the shallow groundwater system. So if you are there's agricultural field here, you will see more nitrate pollution here, so as compared to the deeper groundwater. Okay, so such things, spatial temporal variations are always there, and again, based on your research objective, we have to be thoughtful about all the types of spatial variations that you're supposed to see. And for groundwater also, bedrock is an extremely important factor. Okay, wherever there is change in the bedrock, okay, there will be changes in the water chemistry. So you will have to be aware of the lithological and the anthropogenic usage when you're doing groundwater chemistry. Now, not only water we are collecting, we also need some supporting data. Okay, so we also need to collect those supporting data when we are going to collect for field sampling. So the very basic things, of course, I think everyone will know, is that whenever you're visiting a place, you need to note down the latitude, the longitude and the elevation of the particular place. Okay, so that is called georeferencing of the sample. Okay, so that is extremely important, not only for all samples, but there can be certain samples which actually has a relationship with the latitude and the longitude. So for example, stable water isotopes. Okay, if you're doing stable water isotope analysis, knowing the latitude and the longitude is extremely important, because stable water isotopes vary with the latitude and the longitude of the region. Okay, so you can make inferences therefore based on the stable water isotope variations that you're seeing. Okay, so lat long is extremely important. Date and time, again, extremely important. What is the season that you're collecting? What time is it? Okay, whether it is daytime, whether it is very early in the morning, whether it is, close to the evening, at what time of the day you're collecting a sample is extremely important, and especially for those particular constituents which has, which varies with the organic photosynthetic, cycle. Say for example, DO, dissolved oxygen of the river, if you measure for a river, it becomes slightly different because the river is always moving. Okay, so it might not be as prominent as a stagnant water body. So in a stagnant water body, if you measure the DO with time, you will see definitely a cycle. Okay, so that cycle is related to the photosynthetic activity, right? So during the daytime, sunlight will be there, so there will be more photosynthetic activity, and during the night time, again, there will be less photosynthetic activity. Now related to that, there will be some changes with the DO, and you may see certain changes in some other nutrients, or certain metals also can sometimes show slightly variations in the diurnal circle. Okay, so therefore, whenever you're, based on the parameters that you're going to analyze, you need to know beforehand whether you should take a diurnal circle, at what particular time it is best that you take a sample. Okay, so that is extremely important to note down, right? Discharge. What is the volume of water that is flowing in the river at that particular time is discharge. Okay, now what is the discharge is extremely important, because if it is very high flow, then the concentrations can become diluted, right? So the concentrations will fall. Same if it is, if it is very low flow is going on, then the concentrations may actually be, just because there is evaporation, there is accumulation, right? So there is enrichment due to evaporation. So for that also, the concentration might be changing. So C, that is the concentration, and the discharge relationship is, very different for different rivers, and for every river we may have to find out what is the relation. For that, understanding the discharge and therefore connecting it with the concentration that you are reporting is extremely important. Flux calculation. Flux means what is the total amount that is reaching from the river or from one system to another. Say there are two systems, one is A, one is B. From A to B, what is the total volume of transfer of a particular element is happening? Okay, we generally, we calculate elemental fluxes. Okay, so the total volume in a particular fixed interval, so mostly that is the annual scale. Okay, so the volume of, say, suppose what is the total concentration of calcium is reaching from a particular river to the ocean. Okay, because as I've already said, that rivers drain into ocean, so the ocean is a sink. Okay, rivers are draining into the ocean, so whatever the river is draining, it is all finally going into the oceans, right? So the ocean, which is the final sink for the rivers, what is the, flux of a particular element, say suppose calcium is going from the river to the ocean, we are going to measure that. So for that, understanding the discharge is extremely important. Okay, because in our flux calculations we need the discharge values. Okay, again, for sediment transport, how much sediment is getting transported, that is dependent on the carrying capacity of the river. So whether the river is at high flow stage, whether the river is at low flow stage, whether the river has the capacity to, carry that much amount of sediment, so that is, what is understood from the discharge value. Weathering, whether, from the CQ relations you can make, guesses about how the weathering activities are happening, whether there is continuous weathering activities, you will see from your base cations and your geogenic metals. Okay, so discharge data is extremely important for understanding different kinds of other interpretations. Then there is physical appearance, how the river is looking, right? Whether it is looking like clear, whether it is looking more turbid, whether it is looking green. So that gives us implications. If the river is looking green, that means there is a lot of algae in there. There is a lot of eutrophication related issues can be happening. Okay, so color of the river not only gives us so it indicates important signals about its chemistry. Okay, say for example you're doing a nutrient study. Okay, and while doing a nutrient study you find out the nutrient was very high, that is after analyzing the sample, right? But when you are going into the field itself, if you see that the water is looking very green, you already know that the nutrient load of that particular system is very high because the system looks green. There is a high algal bloom that has happened. So definitely chances of eutrophication and having higher nutrient values will be extremely high. Okay, so therefore you may now again, if the nutrient has all been utilized, then again it is a different issue, but chances will be that if you are seeing a continuous bloom, then nutrient values are getting sustained, and you can make inference that there is maybe some leakage happening from some sewage pipes that is continuously entering into that particular lake or river system, that is changing the nutrient concentration of the system and therefore causing the algal bloom. Okay, so physical appearance is extremely important. Or for example, there will be maybe a flash flood which has happened somewhere there, that is in the mountain regions, and suddenly the water which looks very clear has turned very turbid. So you know that somewhere maybe there is a flash flood which has happened, or there may be a landslide that has happened, due to which the river has suddenly turned so turbid. Okay, so physical appearance is of extreme importance. Then sometimes a lot of floating debris can be seen. You will know that okay, from a particular point source there will be a lot of debris coming, or so such visual, when you go to a particular location, what exactly is happening to the system, you can see the physical appearance and you can say, okay, sometimes you may not even know, so there will be smell, right? If something is decaying, okay, in the river, if the fishes are dying, if there is plants are dying, there will be a different kind of smell. So from that also, from that odor also, you will understand whether the system is a healthy system, whether the system is already compromised, and therefore you're looking at a stressed system. Okay, then what is the natural component of the system? You need to take a note of that. Okay, you need to take a note of the flora and the fauna. Again, this is again based on the kind of research objective of what one is trying to achieve in their study. But if in general you need to know whether that system is a healthy system or not, you look at the flora and the fauna. If you see many species are there, you can see species of all kinds, like there will be birds, there will be insects, not only fishes. Of course, you will definitely try to see if there you can see some fishes or not. So bioassessments are extremely common, and they are extremely fast way of understanding whether system is healthy or not. Okay, so whenever there is a water quality assessments happening, bioassessments, that is ecological assessments, are extremely common. They try to find out how many species are there, okay, how many species of different, different families are there, okay. So based on their flora and fauna, they make an assessment and they say, oh, okay, this particular species is here, that means the water quality is really very good, okay. This species is here, oh, the water quality is really bad here, okay, these kinds of worms are here. So different kind of species gives us understanding of how the health of the system is, okay. And then there are certain key species, okay, that are called the keystone species, whose appearance, gives us confidence that the health of that particular system has improved. For example, you must be knowing that the Gangetic dolphin is one of the keystone species in the Ganga river, and up to wherever it is, seen, okay. So up to wherever it has been seen, we say that, okay, the Ganga river, the health of the Ganga river, the quality of water in the river is good, okay. So if you can spot a dolphin, you can definitely say, okay, the river water quality at that particular location is very good, right. So when the Namami Gange program was going on, and many measures were taken such that pollution can be controlled in the Ganga river, the returning of the Gangetic dolphin up to the midstreams and the upstream locations was a great indicator to show that the water quality is slowly improving, because they cannot live in contaminated or compromised water. So there are certain species in every river, in every ecosystem, which gives us such kind of understanding, such kind of indication. Again, look at the anthropogenic stresses, okay. There can be different types of anthropogenic activities happening. For example, in India, there will be a lot of crematorial grounds on the banks of the rivers due to the religious significance of different rivers. The scenes might be varying on specifically, for the Ganga, that is a sacred river. Many other rivers are also considered extremely sacred: Narmada, Godavari, okay, Kaveri. They are all considered to be sacred rivers, and you will find a lot of religious, ritualistic activities going on the banks of the rivers, and they are not acting as stressors, but they can act as a pathway. So you may think that you are not adding anything like a pollution, you're polluting, but when you see the collective output of it, you will see, oh my god, so much is going on. Like for example, I may have given this example previously, that you're trying to give you're lighting diyas, we are offering flowers, okay. So many kinds of chemicals can get passed through the flowers, through the food, different types of sometimes crops are offered to the river goddess. So from our humans to the river, a lot of things are getting transmitted. So from the ritualistic activities, or maybe due to absolutely anthropogenic, like sometimes a bridge, if a bridge is being constructed. So this is an example from Patna, where over the Ganga river the bridge is getting constructed. So such kind of practices, or you can see a lot of bathing activities in this particular picture that is happening near Kolkata. Floating wastes are very common all over, all over India. You will see there will be rivers, and there will be certain dumping areas close to those areas, rivers, and also there can be subjected to mining, sand mining, right. So sand mining activities are going on. So what they are doing, basically they are collecting the sand. Mostly they will be of illegal nature, and they will be collecting out, digging out more sand than it is permitted. And this activity can actually weaken the banks, and it can be extremely detrimental for fishes because it leads to loss of habitat, and several other aquatic disturbances can happen because the water gets very turbid. There is continuous anthropogenic stress going on, so the aquatic life can actually show that there is like evidences as that activity is happening. Okay, so these other kind of data we need to take a note of when we are going for sampling. Okay, so whenever you take a sampling, you are not just blind, okay, to whatever is going on. It is your surrounding. You take note of whatever you are seeing. Okay, then what kind of instruments you are carrying, right? So there will be certain data set which has to be measured right at the field itself, right? Because if you bring back the sample to the lab and then try to measure that particular parameter, it might change. For example, temperature, right? So if you are putting a thermometer into the lake, if you're putting it into a river where the water is continuously flowing, there the temperature you measure, and then you bring the water, okay, back to your lab, and then you try to measure it. Again, it will be different because your scene has changed, right? Once you were there on the banks of the river was having its own temperature. Now put it in a separate a bottle, you're putting it in a maybe in a jerry can. So then you're bringing it into the lab, and that may take a few days, right? So when you're bringing it back to the lab, wherever that particular lab is, geographical location, based on the weather of that area, your water temperature is supposed to change, right? So certain parameters we measure immediately at the field site itself. So these are called the in situ data set and in situ measurements. Okay, so physiochemical parameters, and they are of physiochemical nature. Okay, so they are basically physical, but again depends sometimes on the chemistry. So temperature, pH, salinity, dissolved oxygen, total dissolved solids, electrical conductivity, resistivity, turbidity, hardness, all these parameters we are going to measure in the field itself, right? So we are going to use different kinds of sensors. So sometimes these sensors are packed into one probe. So it's called a multi-parameter probe. Okay, so this multi-parameter probe, this is a different kind. This is called a pen probe. Okay, so you basically have to collect water in a separate container, and then you're going to dip it. Okay, so this pen probe, you cannot put it directly into the flowing water itself as compared to this kind of probe. Okay, so this has a sensor which is attached to a wire, right? So you can attach this probe directly into the flowing stream. Okay, so this is another different kind of instrument as compared to this. Now this is an automatic system. These are larger probes which you can fit at a particular location, and as the water is flowing through the system, okay, the water flows through this sensors, it starts recording. Okay, it starts recording, and based on the intervals that you have set, it can record data. Say maybe you are recording temperature, and maybe you are recording electrical conductivity, and you are recording turbidity at a particular interval, right? So at particular, say maybe 30 minutes apart, you're recording the data. So at every 30 minutes, at every you'll have a temperature record, you'll have the EC being recorded, you will have the turbidity being recorded. Okay, so these are automatic data loggers. Okay, so they are fixed depending on the bank, and you are taking continuous measurements. Okay, so this is a different nature. Okay, this is a pen probe, smaller item, more suitable for groundwater and all, lake water. This, if you're doing depth study, you need to lower the probe to different depths to see how depth wise changes are happening. Say you want to see how salinity is changing with the depth, how dissolved oxygen is changing with the depth, then you can use these kind of probes where you can lower the probe at a particular depth and see the particular value. Okay, or you can have these kind of automatic systems where you fix an regular interval and data gets logged at that particular interval. And mostly these this has a accumulation of, there will be different kinds of sensors. They will be arranged and there will be one single sensor. Now also, when you're going to take a sample, you have to be extremely mindful of many other parameters. First of all, you need to know what kind of sampling bottles and all that you're carrying. So I already discussed in the, I think in the 8th lecture, that different kinds of analysis requires different kind of bottles and different kinds of preservatives. So you need to have all those ready when you're going for field work. Okay, then we do not collect water from stagnant pools. Okay, so if the water is not flowing, say suppose if there is a lake, okay, and you see that the water there is murky and is filled with insect larvae and all that, and it is absolutely cut off from the main pool, then we do not collect from such standing water, because that is not what the actual system is, okay, that got separated, right? So we are always trying to look at natural water systems. So when we are trying to have a mixed sample, so we are trying to go away from the stressing point where maximum stress is. So suppose you see a bathing ghat or you see a dumping ground, okay, we are not going to take water anywhere close to that place. We are going to the river. If it's a river, we will try to take a sample from the midstream. Okay, you will always prefer to take a boat, okay, go to the middle of the channel, okay, take a water from there. Okay, otherwise wherever you can find a flowing water is there, there you take the water. Okay, be very cognizant that bank activities can change the water. So it is preferable that you use a big rope and bucket and proper precautions so that you do not mix. Many times it so happens that we collect water from a bridge. Okay, so bridge is there and you lower your collecting device from that bridge, so that you're collecting it from the mid channel, or you build such a system that allows you to take water from the middle of the stream and not exactly at the side of the stream. Okay, so you basically you have to do it, build it in such a way that you're collecting the water from the flowing water. Okay, then we never get down and stand in that water. Okay, so whenever we are collecting water, okay, we are collecting it such that we are not contaminating it. Okay, so you have to position yourself as such, if even if you have to get down in the water channel. We always use a stable and firm ground. Okay, so no harm should happen to the person who is collecting a sample. Always remember that there can be currents involved if you're trying to go down into a river stream, or if there is a lake, so there can be currents. So always be very precautious. Always collect samples in duplicate as much as possible. Try to have an extra sample, and they should always be labeled immediately and named properly with the proper sample numbers and date specifically. So these two things are extremely important. One is sample number and the date that you're collecting. Okay, because sometimes it can happen that the date is getting rubbed off, you have only the number, or the number is rubbed off, you have only the date. Okay, so you should use proper methods, use permanent markers, use sellotapes and all that, so that samples are there. And duplicate samples we are collecting, sometimes so happens that some bottles get broken, gets leaked, the sample is lost. So all that kind of precautions has to be taken. Then we always wear gloves, okay? We do not want to contaminate the water, okay? So whenever we are going to collect a sample, we do not want to contaminate the bottle that we are going to collect and store the water. We do not want to uh, have any kind of contamination coming from our side. So as a preventive step, we are doing gloves. We should be always wearing gloves, okay? Then we always should use a life-saving jacket. We should have proper safety gear. We should always have a helping hand to look at when we are trying to sample, okay? So sampling generally happens in groups, okay? If you are going for a river survey, then you will see that you will have people taking you inside the river, so you should have proper life jackets and proper safety gear for your own safety. Then we have to store the samples appropriately. As I've already discussed, that certain samples need freezing, okay? Certain samples need to be stored in the dark, okay? So all these factors should be kept in mind when you're going to take a sample, because in case you're after an entire sample campaign, if you come back and you see, oh, I forgot to freeze these samples, or these samples were supposed to be kept in this way and I had not done it, then that entire campaign, the amount of work you have done there gets just lost. So always when you're preparing for sampling campaign, you should write down what things you should be taking care of. You should have a proper protocol, a sampling protocol should be there with you always, and there should be somebody else also who should be knowing that, okay, so that both of you can always discuss and move ahead. So with this, I come to the end of the water sampling techniques. In the next lecture, we will discuss about the instrumentation, where we are going to, fill in the gap between a sample and the data. Okay, so see you in the next class.